So this is the review of the practice test for the quiz on the scientific method and measurement. What kind of observation would involve subjective or relative values? Okay, well, subjective or relative means it's not something that you're measuring. It's not a measurement. It's not a number necessarily. It's not something you counted. So it's things like a color, like it's blue, or it's hard, or it's big. Those are all things that are not measured. They're qualities of something. So qualities are what we call qualitative observations. The other type of observation you can make is called quantitative, and that is something that you measured. It is 20 centimeters long. It is 15 grams, or it has a wavelength of 690 nanometers. Things like that that you measure or that you count. There are 15 students in the class. That's a counted numerical observation called quantitative, but the answer for this question was qualitative. What factor do you plan to measure in response to a change? So there's two types of variables. There's the independent and the dependent variable. The independent variable, or sometimes called the experimental variable, are the ones that you change on purpose. So you want to change how much water you give a plant, and you want to see if that affects the plant's growth. Well, the thing that you're measuring the effect of, the effect on, in this case, if it was how tall the plants grow, you're measuring what's called the dependent variable. The dependent variable depends on what you're changing. So you change the amount of water and measure how much the plants grow. How much the plants grow is the dependent variable. It's the outcome. You change the way you teach a class, and you measure the student scores. So the student scores are the dependent variable. It's what you're measuring at the end after you've changed around that independent variable that you're testing. The conclusion of an experiment. Okay, so conclusion is one of the steps in the scientific method in the process. And you've had your hypothesis, you've done your experiment, collected data, you've done your analysis of that data, and then based on the analysis, and this got formatted a little strangely, but your conclusion is when you take the results of the experiment and compare it to what you predicted in your hypothesis. So my hypothesis is if I water the plants once a month, then they will grow less. Okay? Then so that's my test. I'm going to water the plants different amounts, and my prediction is if I water the plants less, they will grow less. So I water the plants at different amounts. At the end, I measure all the plants. I look at the pattern if there is one. And if the plants grew less that were watered less, then I compare that to my original prediction, my hypothesis, and I say, okay, this matches my hypothesis, which means the experiment supported, and that's my conclusion. If the plants that were watered less grew more, my conclusion then would be that my hypothesis was not supported. Either way, it's still a valid experiment, but the conclusion is really taking a look back and saying, all right, does what happened match with what I predicted? The experimental procedure that you're testing is what's known as the independent variable, going back to the other question about dependent variable. So the thing that you're testing, and one way to try to think about this is you can always ask a question. That's basically what the hypothesis is, or sometimes called the research question. So you're saying, if I water plants less, then they'll grow less. Well, the if part, if I water plants less, is always the independent variable. The thing that you're going to do on purpose. I'm going to water plants less if I water them less. That's always the independent variable. And if you think of it in an if-then statement, like if I do this, then that, the if, which starts with I, is always the independent variable. And then the then, right? If I water them less, then they will grow less. The then statement is your dependent variable. Then they will grow less, which means I'm going to measure how much they grow. Two groups for comparison. So in many, many types of experiments, and you can't always do this, but ideally what you have are two groups that are treated differently. So one group is basically sort of what you might call the normal group, where nothing is done. If you're doing, plants, you water one plant according to the directions on the plant's care package or care instructions. That would be what's called the control group. It's the one that follows the normal pattern. Then you have another group that you change, like you water it more or less. That's called the experimental group. So the two groups you want to compare are called experimental groups and control groups. The control group is the group that's unchanged, and then the experimental group is the one that you play around with, and then you compare the two, because if the experimental group and the control group are different, then that means the thing you changed in the experimental group had some effect on that group, or at least that's what it would seem to suggest. The information you gather from observation is called data, right? And again, we said there are two different kinds. There are measurements, which is quantitative data, and then there are observations of qualities and things, which are called qualitative. So it was bubbly. That's a qualitative statement. It was 72 degrees Celsius. That's a quantitative piece of data. But they're both data, and those are the observations you make during an experiment. The final step in the scientific method is to share your results. Okay, part of the progress of science is based on scientists doing experiments and then sharing their findings with other scientists who can then either build on that work or look at it and maybe find a flaw in the work that could be improved upon. But all this is dependent on sharing your work with others so that they can find out what you're doing and they share their work back with you so you can see what they did. So we're not all trying to do the same thing at the same time and in that sense kind of wasting time. But we can build on each other's work or find better ways to do things. What's the first problem? The first step is the problem, right? The first step in the scientific method is seeing something that you don't understand, that you can't explain. And then the scientific method is the process of trying to come up with an explanation. So the very first thing you have to have is something you don't understand. If you understand it, you don't need to go through the scientific method to explain it because you already understand how it works. Why, or which is not a reason why we use the scientific method? Well, certainly not because Mr. Scott says so. The results being multiples of 10 is not about the scientific method. Results will only be published. That's not really why we use it. It might be a true statement, but that's not why we use the scientific method. And it would be understood by everyone except U.S., Liberia, and Myanmar. That's more about the metric system versus the English system. So the answer is none of these are reasons why we use the scientific method. We use the scientific method because it's a repeatable, easy-to-follow process that produces valuable information related to problems that we don't understand. And this whole 2 lines here should have been erased. So the question was, a data table is used to blank. Well, a data table is used to collect your observations. All right, you're measuring things during an experiment. You write down either your qualitative observations or your measurements. You put all that into a table so that it's easy to follow later on, and you can track everything. You have a table that might have spaces for each trial that you do an experiment. You might have it spaced out by certain times during the experiment, but it's important that you have a table set up before you start that will be able to collect all your data so that it's organized as you're doing it, and then you can refer back to it later and it's easy to follow and understand and see what was happening during the experiment. Okay, so I'm not going to read through the entire experiment, but we have this student who was trying to find out about the effect of acid rain on pine trees. So the question is, which of the following is a quantitative observation? Well, the only quantitative, again, you have to remember quantitative means measured or counted. Well, so the only one of these that's a measured quantity or a measured piece of data is the number of trees she selected. There are 6 trees in her sample. All the others are kind of observations. The color, the needles were curled. Well, again, that's sort of descriptive, but it's not, measuring the angle of the curl or the arc of the curl. And the acid trees looked sick. Well, again, that's more of a matter of opinion. Some people might think they do, some people might think they don't. That's what makes it qualitative, not quantitative. Which is not a piece of data that Brenna collected. So remember, data is observations made during the experiment. So either whether they're measured or not, they have to be things that you observe during the experiment. And she observed all of these with the exception of sulfuric acid is not an observation that she made during the experiment. It may be something that she used in the experiment, but just the compound sulfuric acid is not an observation in itself. What was the problem her experiment was designed to investigate? And again, the question that she was trying to answer is, what is the effect of acid rain on pine tree growth? Okay, that was her experiment. She used different amounts of acid and measured how much the trees grew. So the independent variable was the amount of acid, and the dependent variable was how much they grew. So again, the effect of acid rain, that's the independent variable, on the dependent variable. So independent, dependent. Which variables were held constant? Well, I guess you could have selected 2 choices here. The amount of sun received, it was said that she put them all next to each other, so at least theoretically they should have all gotten the same amount of sunlight if they're sitting right next to each other and not being blocked by a roof or a bigger tree or something. And technically you could say that the large red pots were constant. It's not really a variable, but I guess you could argue if she used different size pots or something that maybe that affected the growth as well. So either one of those would have been a correct answer there. What was the dependent variable? Kind of talked about this a couple of questions ago. The thing that was measured at the end of the experiment was how tall the trees grew. So she's measuring the effect of the acid rain on the height of the trees. So the height of the trees is the dependent variable that she's measuring at the end to find out if there was effect. Which trees would be called the experimental group? Well, the experimental group is the one that you change something. So the ones that got water would be what we call the control group. So they're kind of getting the normal condition. And then the trees, all of the trees, even though some of them had different amounts, all of the trees that got acid added to the water would be considered experimental because you're changing the stuff that they're getting. What was the independent variable? The independent variable that was changed was the amount of acid in the water for the different groups of trees. So that's the independent. The dependent is how high they grew. The independent is the amount of acid to see if there was any effect. What could Brenda do to further investigate? Well, really, she could do any of these. All of these would be very valuable ways to gather more information about her hypothesis. She could repeat her experiment exactly the same way because even though when she did the experiment it looked like the acid shortened the trees, maybe there was something else that she didn't have control over that had an effect as well. If she did it again, or even better, several more times, then she would be able to say, all right, I did this 10 times with 100 different trees or 100 individual pine trees, and every time I added acid to the water, the trees didn't grow as much. That would be a much more valuable statement than saying, I took a few trees, I added some acid, the ones with the acid didn't grow. Now that's a good experiment, but repeating it over and over adds value to your conclusion. Repeating the experiment with a different independent variable would also be valuable because maybe it wasn't the acid, or maybe the acid didn't have an effect and it just was a combination of things. So if you go back and you do the experiment again, but this time you change, say, the amount of sunlight, and you hold the amount of acid constant, maybe that will show you something different. And then, as we said earlier, a very important step in the process is sharing your work so that they can observe her observations and take a look at her results. So all of the above was the correct answer here. Okay, so we're going on to the measurement section. Okay, the basic unit of volume in the SI system, you should know all the basic units. Liter is for volume. Time in the international system is measured in seconds. Energy is measured in joules. So this is, gram is for mass, meter is for length or distance, liter is for volume, joule is for energy, second is for time, and then the other ones are, we use temperature, which is either the Celsius or the Kelvin scale. The prefix 1 that represents 1 million is, and actually there was a mistake here, it's actually none of these. The correct answer is mega. Mega means million. Okay, megabytes, megaliters means millions. The representative of 0.01 or 1/100th is centi. So you need to know, you really should know the prefixes from kilo to milli. So kilo, hecto, deca, base unit, deci, centi, milli. If we convert meters to kilometers, so if you have kilo, hecta, deca, base unit, deci, centi, milli, you move along this line, the number of places you jump is the number of times you move the decimal. So going from base unit, which is meters, 1, 2, 3 places to the left, to get kilo would give you an answer of 0.0689. Grams to milligrams, so base unit to milli is 3 places to the right. So we would add 3 more zeros, that would be answer choice E. Decaliters to centiliters, so now we're going, so kilo, hecta, deca, base unit, deci, centi, milli. So if we go from deca, 1, 2, 3, to centi, we'd have to move it 3 places to the right, and so that would be answer choice E again. Number 9, kilometers to millimeters. This time we're going 6 places to the right, so we're going to add 6 zeros there. Answer choice D. Meters to millimeters. All right, so that's base unit, deci, centi, milli. 1, 2, 3 places would be 8,730. Milligrams to grams. Now we're going from milli, 1, 2, 3 places to base unit. So 1, 2, 3 places to the left gives you 0.00254. Milliliters to liters. Again, now we're going 3 places to the left. So 1, 2, 3 gives you 0.214 liters. Centiliters to centimeters rather to kilometers. So we're going from centi to kilo. 1, 2, 3, 4, 5 places to the left. So that would mean, since we had 1 zero in the decimal there, we'd need 6 zeros. So 0.0000079. 17.5 millimeters or milliliters to centiliters. This one actually had a typo as well. It's none of these because we're only going 1 place. So it would be 1.75 centiliters. The length of the line below. This is really just a matter of having some idea of the difference of scale between say so this is nanometers, kilometers, centimeters, millimeters, and gigameters. Well, giga is a big number, that's a million. Kilo is 1,000 meters. So a meter is about a yard for, to convert it into something roughly what we're used to. So a kilometer is 1,000 of those. So that's really far. That's like a little more than half a mile. Which means gigameters is way, way, way more. And then nanometers, nano means really small. So really, really small piece of a meter. Millimeters is 1,000th. And a millimeter is not very big at all. Centimeter is about half an inch. Again, converting it into units we're familiar with. So a centimeter is about that big. So this is probably close to 14 centimeters because the others are either way too big or way too small. Why is the SI system considered easy to use? Well, the correct answer here is that base units are connected to phenomena that are accessible by everybody. So as opposed to the old system where, first of all, they either weren't common units or they were common standard units, but they were, say, like we talked about the king's foot being the measurement of a foot. Well, not everybody can get to the king's foot. They just weren't allowed to be near the king. So the fact that these are natural phenomena that everybody can see means anybody has access to the basic information. The other things that are make it useful are it's multiples of 10, which is easy to remember. All of the units have the same base unit. Different measurements have different base units, but each base unit can be added to by different prefixes, which makes it easier to follow because a kilometer and a meter are measuring the same thing because they both have meter in them. It's just that kilometer is a different number. Whereas in the English system, you've got miles and furlongs and fathoms, which are all measurements of distance, but you would have no way of knowing that they measure the same thing because they're not related. The words themselves are not really related. Which of these is an SI measurement? Well, the only one that's part of the metric system would be this decameter, because it's got meter in the name, which is the international measurement for distance. All the other ones, ounces, miles, hours, and furlongs, are English measures that we use here in the United States. In the prefix metric system, 1/1000th is called millimeters, milliliters, milliseconds. And multiples of 100 is hecto. Sorry, not deca, hecto. Centi would be 100th, so 1 over 100, but 100 things is a hecto. So this isn't one that gets used a lot, but hectares is actually a measurement of area. It's like acres, but in the metric system. So it's just not one that's used a whole lot, but we would use it if we were measuring something in hundreds. 1/10 is deci, like decimal, right? The decimal is measurements in tenths. And that is the last question on the practice test. So there will be a couple of questions that weren't on here, or a couple more questions related to the scientific method specifically. It's just the questions on the test can't really be asked in a different way. So there will be 2 more questions on the scientific method, but if you're studying all of the questions on here and you understand the concepts of the different types of variables, quantitative, qualitative, the steps of the scientific method, and things like experimental and control groups, you'll have no trouble answering those extra 2 questions because they're still about the scientific method. Okay, so that's it. You need to make sure you know at least this part of the metric number line, right? Kilo, hecto, deca, base unit, deci, centi, milli, so that you can move back and forth between the units or between the prefixes easily. And I think that should be it. So good luck, and we'll see you for the quiz.